Hi everyone, I'm Dr. Fizz, and let's learn some physiology. There's no intro today. I'm not going to be editing this video, so you're going to get the raw, uncut version of Dr. Fizz's videos. I don't have time to edit them. But today we are going to go through the neuromuscular junction and skeletal muscles, and I'm going to use my notes here to draw on the PowerPoint, and we're going to get right to it. All right, so breaking this down, what we want to cover today are the molecular characteristics of the sarcomere, or the muscles and the muscle cells as a whole, mechanism by which calcium is released from the sarcoplasmic reticulum, as well as how muscles are contracted, and what energy they use for muscle contraction. All right, so breaking down the drawing here, there are 1, 2, 3, 4, 5, or 6-ish different parts to this. We'll start with the entire muscle as a chunk. So you can imagine that this would be your bicep that you have on your arm, hopefully. This is the entirety of the bicep. It's covered by something called fascia, and on the underneath, deep to that fascia, is called the epimysium. The epimysium is another covering that separates the fascia from the actual muscle. Going even deeper than that, you'll see these little lines here that represent this big thing, which is called paramecium. Paramecium covers something here, which are bundles of muscle fibers that are wrapped together in something called the endomysium. The endomysium keeps all of these muscle fibers together, and each of these little muscle fibers are technically muscle cells. So breaking down muscle cells, you have the sarcomere that covers the outside of the muscle cells, as well as something called dystrophin, which links the sarcolemma, which is the outside of the cell, to the endomysium. On these little bubbles on the outside are on the nuclei of the cell, and inside you can find squashed between something called myofibrils. You'll find the mitochondria, which is the powerhouse of the cell. Deeper than that, these little bundles right here that extend out this is called, like I said, the myofibril. From this is made up of actin filaments and myosin, and we're going to go deeper into that. All right, so I mentioned before the word sarcomere. This we had mentioned just briefly, where the sarcolemma covers each muscle fiber or cell, and this is the structure of a sarcomere. So we've got the covering, sort of the fascia-like structure on top of the muscle cell. Each of these have invaginations, and we'll talk about what each of these are. So these are called T-tubules, basically just sarcolemma extended, but to the point where they're invaginations and have contact with the walls of what's called the terminal cisternae, and protruding from the terminal cisternae are the sarcoplasmic reticulum. Long story short, the sarcoplasmic reticulum allows for the transfer of calcium and can be released into the sarcoplasm, which is basically the cytoplasm of the cell, when an action potential hits. So let's go deeper than that. First, this three system area here with two terminal cisternae and T-tubules are important. This is called the triad, and we're going to go deeper into that. And the junctions between here are going to be essential for actual contraction of muscles. All right, so looking at actin and myosin, this is what we saw earlier in our slides, where they protrude from the myofibrils. So these are the two molecules that are, or compounds, I should say, that are super essential in muscle contraction. So right here, breaking down this thing called myosin. I'm sure you've heard of it before. It's also called the thick filament. And we'll break that down even further. But long story short, it has a tail, which is right here connecting to the main chain filament. Each of these have a myosin head, which are going to be super important because they interact with something called the actin, or thin F-actin or thin filament. The F-actin is made up of three different major compounds, which is G-actin, these red circles here that intertwine. Four compounds. They intertwine and surround something called nebulin, which holds the whole complex together and makes it a straight line. Tryptomyosin in this purple line right here can hold about 7 or 8 G-actin proteins together and have them all interact. And the thing that actually connects each of them are these green little 3 circle areas over here called troponin. Troponin is made up of 3 different parts. Troponin I, or 1, I'm not really sure what it is, troponin C, and troponin T. Troponin 1 connects actin to troponin. Troponin C has 4 different calcium sites, 2 which are high affinity and 2 which are low affinity. And then troponin T connects actin, and therefore actin to tropomyosin, and troponin T and troponin C are very closely related. That when calcium binds and myosin will bind to the active sites on G-actin, a conformational shift will happen here that causes tropomyosin connected to troponin T to shift and allow these myosin heads to connect. So let's talk about that. All right. So as I mentioned, we have actin and myosin, and we're going to talk about how muscles contract. So as you can see here, this is the structure of the muscle. You have myosin here. Myosin heads point left, and myosin heads over here point right. They're connected to actin and other molecules through something called titin, specifically connected to something called the Z caps right here. And let's see, the Z caps and those are connected here, as you can see. Nebulin also anchors into Z caps, and this is called as a whole the Z disc here. You might have seen in our previous slide, you're welcome to go back and look, how there is a black zigzag line on the myofibril, sorry. Those are technically Z discs, and those kind of separate the areas within the muscle that physically contract. So those are super important. And this area that physically contracts is called a sarcomere. So this is like one little unit, and it keeps going like this on each side. All right. So looking at it as a whole, you have the M line, which divides myosin right here, actin, Z caps, the Z disc as a whole, connected to more nebulin, which is surrounded by G or F actin. Looking down here, I mentioned earlier the actual contraption about how muscles move. It almost acts like an accordion, but more, I guess you should say, pistons in a car. As titin sort of anchors the myosin to the actin, G-actin has specific binding sites that are allowed to activate when calcium binds to troponin C, causing a conformational shift where tropomyosin will move, exposing the binding sites, and then myosin heads will attach there, break off through a system we'll talk about later, and then attach to new ones. And this will cause, like an accordion, or a piston, I should say, like a piston, the muscle to contract and then return to normal. Alrighty, so talking about how calcium is released in the sarcoplasmic reticulum. We've talked about in previous video series where something called a signal will come down the neuron, will open up and create an action potential, opening up calcium channels here and other channels. Calcium channels allowing these purple dots, these calcium, to enter in. Clips snare proteins and then allows those snare proteins to attach to the axon terminal here. The axon terminal will then allow it to these vesicles that are full of neurotransmitters to release into the synaptic cleft, and that will release neurotransmitters, specifically in this case acetylcholine. Let me catch up with my notes real quick. So release acetylcholine into synaptic cleft, which acetylcholine will then bind to the acetylcholine receptors represented here in an orangish brown, which are also nicotinic 1 receptors or channels. And these are found in specifically on the neuromuscular junction. So N1 is always found on the skeletal muscles on the sarcolemma. So this right here is specifically the sarcolemma, which is, you could say, the cell membrane almost of the cell. Let's see. This is also called the motor end plate. Anyway, so the attachment of acetylcholine to these will actually open up these channels and allow ions to flow in, which these ions are sodium, for example. And this will cause a change in action potential. I guess they'll let sodium ions in through these little channels, which will change the membrane potential, opening up more voltage-gated channels, and thus creating an even bigger action potential in the cell. And we'll come back to the motor end plate in just a bit, but with the membrane potential continuously sending a signal down, we talked about how what are they called? The invaginations there, also called T-tubules on the sarcolemma, will go down deeper along the walls of the cell, and this will allow for more channels to open and for something called the conformational shift to occur on what's called dihydropyridine or L-type calcium. These aren't actually channels, but they perform conformational shifts, which allow for RYR, which is a channel as well that's connected to the sarcoplasmic reticulum, which is full of calcium. The DHP will change, opening up a way for RYR to allow calcium out of the sarcoplasmic reticulum. And we mentioned earlier why that's important. We'll go into that in just a bit. But going back to the motor end plate, the question is what happens if we just have a perpetual acetylcholine triggering these and causing action potentials? Well, that's why we have acetylcholine esterase here, which will actually clip acetylcholine into 2 different molecules, choline and an acetyl group, and the cell will actually intake choline again to make new acetylcholine, which is a neurotransmitter, and the acetyl groups will just diffuse into the space in the body and get cycled out. All right, so going back to the calcium here being released, let's talk about why that's important. All right, so let's talk about something called the cross-bridge cycle. The cross-bridge cycle is important because it shows us how myosin physically attaches to actin, allowing for muscle contraction to occur. So we see here we have myosin with ADP and PI connected to it. As we go through the system, we'll see that calcium that's released from the sarcoplasmic reticulum attaches to troponin C, which allows for a conformational shift in the cell, or in a conformational shift on the troponin, which pushes tropomyosin away from G-actin, exposing its binding sites, and pow, they will connect. So at that point, we have ADP and PI strongly connected to this myosin head, holding it in an upright position. But something called a power stroke can happen, where ADP and PI are thrown off of this molecule, causing it to go back into a stuck state. When ATP comes in, ATP will actually allow it to disconnect, and two different things can occur. This is called the cross bridge cycle, because depending on what molecules are in existence here, we can actually have, well, long story short, hydrolysis will occur, causing ADP to split into ATP ATP to split into ADP and PI again, leaving us back at the beginning. So let's keep going there. We're going to talk about the cross bridge cycle actually later. All right. So there's different ways that muscle contracture can occur. Muscle contracture is the shortening of a muscle and that resists relaxing back to its normal resting length right at the beginning. But the reason that long-term or permanent contractions can happen is if we're, sleeping at our desk or something like that for a long period of time, which are other prolonged immobilization. If we have spasms or spasticity, and or we have muscle weakness, like me there, unfortunately. All right, so there's different sources for muscle contraction. The first source of muscle contraction energy comes from cytosolic ATP, which is basically just a floating pool of ATP in our sarcoplasm. It exists in the sarcoplasm, but it's immediately available. It lasts for about 1 to 5 seconds when using this ATP, or this source of this ATP, but it allows for super fast activity. Unfortunately, it doesn't last for very long, and it's not good for long-term contraction. Over here, we have creatine phosphate, which kind of acts as a backup to cytosolic ATP. It requires an enzyme that's called creatine kinase on the M line that we talked about earlier, and it's used after cytosolic ATP. It's available with this enzyme, and it will decrease energy of activation. So after we used up all of this, then the last little bit of energy will push it. After 1 to 5 seconds of this, about 10 seconds will last of the use of creatine phosphate. So it moves phosphate from ADP to ATP to allow for more ATP to act in that fourth step in that third step of the cross bridge cycle without oxygen, so in an anaerobic state. There isn't a lot of this in the sarcoplasm, so it's not going to be super helpful. It isn't sustainable. All right, moving on. We have something called glycolysis, represented by this sugar molecule, and it's available with how much glycogen we have in the muscle. It depends on our sugar intake. It's a major source of ATP during anaerobic respiration. And it's very quick for intense activity, and this guy lasts. It's different from cytosolic ATP, but this guy lasts for a whole minute, so 60 seconds compared to 1 to 5 seconds. It can be very useful. It's not as powerful, and it's super short, so it's even shorter than this. All right, moving on. When all of those fails, or we're exercising for a long time, we have something called aerobic oxidative respiration, which uses oxygen to produce ATP. It's available wherever oxygen is in the system, and it produces the most ATP out of everything. I believe the whole process, both of these combined, will end up producing about 36 to 38 ATP per cycle, and it cycles millions of times. So it's said that we burn our weight in ATP every day. That's how much ATP, and these are microscopic molecules, so you can imagine how much that is. All right, so it's slower than anaerobic, these guys, And its primary choice is fatty acid, but it's not really it's not for super intense or fast activity. That's what these guys are for. Alrighty, let's see. Let's just go into a review real quick. So reviewing what we talked about nebulin and actin, myosin, troponin, Z-caps, etc. When it comes to the sarcomere and the muscle cells in general. We've talked about how calcium is released from the sarcoplasmic reticulum on gated channels and performing conformational shifts. We talked about cross-bridge cycles, and we talked about the sources of muscle contraction. So with that, I'll leave you with that. Please feel free to talk to me if there's anything that I missed. And yeah, have a great day. Stay busy.